Hello, welcome to Revise with Mwalimoo. My name is Teacher Mwalimoo. Great to have you here. In this session, we will do 2 experiments about the diffusion process. What happens in diffusion? We'll start with a brief definition of what diffusion is. In diffusion, particles move back and forth in any direction from where they are many to where they are fewer. The movement of particles may continue until the number of particles is the same in a space. So basically, what we're trying to say is diffusion involves movement of particles, and these particles move from an area of high concentration to an area of low concentration. Let us watch a brief video that will demonstrate what we have just talked about and define diffusion in a good way.
Diffusion is the net movement of a substance from a region of high concentration to a region of low concentration. Let's use this sugar cube and beaker of water as an example. When the sugar cube is dropped into water, it begins to dissolve. The sugar molecules in the cube move from an area of high concentration to areas of lower concentration until the molecules are evenly distributed. This type of motion is known as diffusion.
Now that we have known what diffusion is, let us go to how we can see this. The process of diffusion may be demonstrated by the following. We can observe the spread of food color particles in gel or dyes from flower petals, drops in gel, potassium permanganate crystals in water, methylene blue drops in gel. That is how we can demonstrate diffusion. So we are going to require some materials that will help us in the process of demonstrating diffusion. One, we will need a clear petri dish, glass petri dish as seen in this image. We will also require a white background. We will require either of the following dye, food coloring, potassium permanganate, or methylene blue in the experiment. Either one of that will be required in the process. As we can see, we have the dye there. That is how it looks. Methylene blue. We have potassium permanganate, and we also have the food coloring. So any one of those requirements will be needed in the process. In this case, we are going to use methylene blue. We will also need gelatin or jello crystals. That is how it looks like. We will need a ruler showing millimeter readings, graph paper with millimeters reading. So in this case, you can see the ruler with the millimeters reading. We will also require a means of timing. You can use your phone, your stopwatch, or clock. In this case, we are going to use this stopwatch. We will also require a dropper for the experiment. So those are the materials that we will need for the experiment. Now let's go to the procedure of the whole experiment. Remember, the aim of the experiment is to demonstrate diffusion using blue food color, that is if you are at home, or methylene blue if you are doing this in a laboratory. Let's go to step 1 and 2. In step 1, You will be required to place the gelatin in a glass petri dish, the first one that we saw, that one that is made of glass. As you are doing this, ask yourself, why is gelatin used instead of water? In this whole process of diffusion, we have decided to use gelatin. Why are we using gelatin instead of water? Very gently add a drop of the blue food coloring. That is if you are at home and you're using the food coloring and also the gelatin. Or if you're in a laboratory setting, you will use methylene blue. So very gently add a drop of either at the center of the petri dish with the gelatin. The drop should be on the surface of the gelatin to avoid a splashing effect. After you have placed the drop in the gelatin that is in the petri dish, you will be required to start the stopwatch that we also put as one of the requirements. Step 3 will be required to place a 30-centimeter ruler along the side of the petri dish as shown in the image below. Remember, we also put ruler as one of the requirements. So we will be required to place a clear 30-centimeter ruler along the side of the petri dish as shown in the image below. In step 4, we'll be required to record the reading of the size of the drop of blue food coloring or methylene blue at the start of the experiment and 30-minute intervals to complete the table below. So you will be watching whatever you have put there as the experiment at intervals of 30 minutes, and you will be recording the reading as you have seen in the ruler, using the ruler. You will be required to record what you see and measure the diameter. Diameter in this case is the distance across the blue circle of the blue color spread. This is what you will be expected to see. You will be expected to see the following. The blue drop at 5 minutes, it was small. At 30 minutes, it started becoming large. And at 60 minutes, we are seeing it is larger compared to the 5 minutes and the 30 minutes. What else do you observe about the blue circle? If your answer was the color is intense at the epicenter, then gradually becomes lighter towards the edge, then you are correct. As you can see from the images, the color is intense at the center, but when we move to the edges, it becomes a little bit lighter. This demonstrates the movement of the particles. After observing, this is the data that you should have recorded. In 0 minutes, we saw, you could have seen that the blue color spread in millimeters was 5. After 30 minutes, it moved to 10. After 60, it moved to 15 millimeters. And after 90 minutes, it moved to 20 millimeters. After 120 minutes, it moved to 25 millimeters. So we have seen the observations. Now the next question might be, how do we explain this? The molecules of the blue food coloring moved from the point of placement towards the edge of the petri dish because of diffusion. Remember, we said diffusion is a movement, and we have seen in the experiment, the molecules moved from the point of placement towards the edge. Therefore, they were moving along the concentration gradient from where they were many to where they were fewer or none. Remember, we asked a question: why did we use gelatin instead of water? If your answer was to reduce the possibility of blue food coloring molecules because of water currents, gelatin has a higher viscosity than water and consequently will not have currents flow inside it. Gelatin, just like gel, it is very heavy. That's why we say it has a high viscosity. So movement is usually restricted, unlike water, which is less viscous. In conclusion, we can conclude that diffusion results with the molecules' movement from regions where they are many to where they are fewer. So we have seen the experiment, we have recorded the observations. Now let's go to generalization of the findings. If you were to repeat the experiment using potassium permanganate, would you expect the same results? Remember, in this case, we have used methylene blue. So if we were to use potassium permanganate, would the results be the same? The answer is yes, because diffusion occurs with all molecules and particles. What more information will you need to compare the rate of diffusion of food coloring and potassium permanganate? The molecule size or molecular weight of blue food coloring and potassium permanganate. The heavier molecule would move slower, hence a lower diffusion rate. So let us watch a brief video showing the use of potassium permanganate in water. We have that involves potassium permanganate and water. We are going to the next experiment. There is a brief background information to the next experiment. So in this experiment, we are going to require some materials, but first let us just explain the background towards this experiment. When iodine solution is added to starch solution, starch molecules react with iodine ions to form a blue-black color. Whisking tubing is a selective artificial membrane that will allow small molecules or ions to pass while preventing large size molecules from passing through. So here we see we are going to need a whisking tubing, we are going to need iodine solution, and we're also going to use starch. But let us just go to the requirements one by one. We are going to need 2 pieces of whisking tubing, which are 10 centimeters each, as seen in this diagram. We are going to need potassium iodide or iodine solution, which is 200 millimeters, as seen in this image. We are going to need starch solution, 200 ml also. We will need 2 clear glass, 200 ml, which are the 2 clear glass beaker, each 200 ml, as seen in the diagram. We will also need 2 glass rods, as seen in the diagram. We will need a thread or string 50 centimeters long. We will also require a white background in this experiment. We will also need a wash bottle for the experiment to be complete. Remember, the aim of the experiment is to demonstrate diffusion using starch and iodine solution. Let us go to the first step. You will be required to place iodine solution in a 200 ml beaker to 180 ml mark and label it setup A, as seen in this diagram. So from this, we are seeing the solution has been placed in the beaker to 180 ml mark, and we will label it setup A. In step 2, you will be required to tie one end of the whisking tubing. We are using the first whisking tubing. You'll be required to add 5 ml of starch solution into the whisking tubing. Take care not to spill on the sides. After adding the 5 ml starch solution into the whisking tubing, you will be required to tie the second end of the whisking tubing, leaving a portion of the string free to tie on the glass rod. So when you are tying the last end, you will be required to leave a portion of the string which will be used to be tied onto the glass rod. You will then be required to lower the whisking tubing into the iodine solution in the beaker to be fully immersed in the iodine solution as shown in the diagram below. As you can see now, the whisking tubing is fully immersed into the iodine solution that is in the beaker. In step 4, you will be required to repeat step 1 to 5, but with the iodine solution in the whisking tubing. Remember in the first setup, we were using the starch solution was the one in the whisking tubing, but in setup B, we will be required to have the iodine solution now in the whisking tubing, and the starch will be in the beaker. You will label this as setup B as shown in the diagram below. We have seen the procedure. Now let us go to the observations after successfully completing the procedure. The observations that we are going to state are both for setup A and setup B. For setup A, in setup A, the color of starch inside the whisking changed to blue-black, as we can see in that image. We have the iodine solution are in the beaker, but in the whisking tubing, we are seeing a change of color to blue. black. In setup B, the color of starch in the beaker and outside the whisking tubing changed to blue-black. So this is what is expected to be seen after successfully completing the procedure. Now let us explain the observations both in setup A and setup B. In setup A, the color of starch inside the whisking changed to blue-black. Why? This is because the iodine ions diffused into the whisking tubing to react with starch molecules. In setup B, the color of starch in the beaker and outside the whisking tubing changed to blue-black because the iodine ions diffused out of the whisking tubing to react with starch molecules. This is, as we can see from the image here. So it is right to conclude that iodine ions are smaller than starch molecules, therefore passing through the pores of the whisking tubing. So let us go to generalization of the findings. What part of a cell was represented by the whisking tubing? The answer here is cell membrane. So you might ask yourself, why is whisking tubing being compared to cell membrane? This brings us to the next question. What property does the whisking tubing and cell membrane have in common? They are both selectively permeable, allowing small molecules to pass through while stopping the larger molecules. We should underline the part selectively permeable. It means it only allows certain molecules to pass and not all. And in this case, the molecules that usually pass are the small molecules and not the larger molecules, as we have seen in the experiment. How does selective permeability affect the diffusion process? Diffusion through a selectively permeable membrane will be possible if the molecules are small enough to pass through the pores. The larger molecules will not pass through. We have seen this in the experiment. Today we have learned the following. Lessons on diffusion demonstration. Diffusion is the movement of molecules or particles from where they are many to where they are few until an equilibrium is reached. So we have seen that diffusion is a movement, yes, and what is moving is the molecules or the particles from where they are many, that is where they are most concentrated, to where they are few until an equilibrium is reached. Molecules fuse faster than heavier molecules. Molecules which are smaller in size will be allowed to move through a selective membrane, while larger molecules are stopped. Remember, the membrane is selective, so it chooses the smaller particles in size and leaves behind the larger molecules. If you have a question, please feel free to ask via the Q&A section. Just click on Q&A and type in your question. we will be able to respond to it. So we have learned how diffusion takes place. We have seen its movement in the two experiments that we have seen, and we have made conclusions. Now let us move to the next segment, which is the revision bit and the type of questions that are usually asked in KCSE in regard to diffusion demonstration. So a group of students set up an experiment to demonstrate a certain process. The experimental setup as shown in the diagram below. After 10 minutes, the students recorded the observation in a table as shown below. Observations inside the tube and outside the tube. So inside the tube, there was blue-black color. Outside the tube, there was no color change. In number 2, inside the tube, there was no color change, but outside the tube, there was the blue-black color. Question 1A. State the process being demonstrated in the experiment. Selective permeability or diffusion. You might ask yourself, why are we saying it is diffusion and not osmosis? Note, because of the students associating visking tubing to osmosis, they give the wrong answer to this question every time it is asked. Therefore, be very careful answering such questions. Whenever you see a visking tubing as a student, the first thing that comes to your mind is osmosis. But in this case, what they're trying to demonstrate is diffusion. So be very careful when answering such a question. In question 1B, explain the results in experiment setup 1. Remember, we saw color change in the visking tubing, and another one we saw a color change around the visking tubing that is in the beaker. So visking tubing is semi-permeable, therefore allows passage of smaller iodine molecules from the beaker into the visking tubing, while preventing passage of the larger starch molecules. When comparing the molecules of starch and the molecules of iodine, we get to see starch has very big molecules compared to iodine. So we are seeing movement of smaller iodine molecules from the beaker into the visking tubing, while preventing passage of the larger starch molecules. So this will result to the formation of the blue-black color with starch solution. Starch molecules are too large to pass through the walls of the visking tubing into solution in the beaker. Therefore, the iodine solution does not change color. Remember, semi-permeability only allows the small particles to move and not the large particles. When we compare starch solution and iodine solution, we get to see that iodine solution has small molecules compared to starch solution, which has big molecules. Therefore, the iodine solution will be the one moving and the starch solution will remain constant. So when the iodine solution moves, it means the color of the starch solution will change to blue-black. Let's go to an example of question 2, as we usually see in the KCSE past papers. So the first one, and a very obvious question, is what is diffusion? The best way to answer this is to say that diffusion is the movement of molecules or particles from a region of high concentration to a region of low concentration. Question 2b of the same question 2. How do the following factors affect the rate of diffusion? Factor number 1 is diffusion gradient. High diffusion gradients have a faster rate of diffusion. Factor number 2 is surface area to volume ratio. Larger surface area to volume ratio has faster rates of diffusion compared to smaller surface area to volume ratio. Important to note, this is the reason single-celled organisms use diffusion. The factor number 3 is temperature. Increasing temperature increases the rate of diffusion. You might ask yourself why. So it is very important to note that this is because temperature increases the kinetic energy of the molecules. If temperature increases the kinetic energy of the molecules, this will mean the rate of diffusion will be high. So if you put side by side an experiment that has low temperature and the other one that has high temperature, and put in potassium permanganate or put in the methylene blue or the dye or the food color, you will get to see that the rate of diffusion in the beaker or in the petri dish that has high temperature will be high compared to the other petri dish or beaker that has solution of low temperature. And as we have explained, this is because temperature increases the kinetic energy of the molecules. Therefore, the molecules will be moving at a faster rate. We have seen how experiment 1 has been done. We have seen the observations. We have gone to experiment 2, the procedure, the observations, and we have given a summary of the lesson, as well as the questions, the possible questions that have been asked in the past KCSE. So now we are going to the next segment, which is the assignment bit. Remember, if you have any question, you can type it in the Q&A part, and we will be able to respond to it. So now it's time for the assignment to help us understand the concepts we have learned better. We are going to do the assignment in Shupavu 291. So we are going to send the word home to 20851. This is using your phone. You will enter the words change subject and pick on biology. After you have picked the biology subject, you will be required to enter the words physiological processes. From there, you will get a lesson. We call it a tutorial. And then after reading through the tutorial, you will be required to answer the 5 questions that come after it. You can also go to Shupavu web by going to www.mwalimoo.com. And then from there, you will be required to pick on biology subject. After you've picked biology subject, you will search for the words physiological processes. Then from there, you will be required to take lessons and questions based on that topic. After successfully doing this, you will be good to go on the topic of diffusion demonstration that usually happens in cell physiology. I hope you had a wonderful session. Kindly give us feedback on how the lesson was conducted by filling the survey. See you in the next lesson.